Hello, in the previous session we have discussed about the classical theory of transpiration, that is the star-sugar hypothesis, and we have also discussed that why this theory has been rejected. In this session we are going to talk about the most accepted theory, and that is the active potassium ion theory. This theory was given by Levitt, right? So one by one we will discuss about it. First of all, let's discuss the sequence of event that is going to happen during the light. So during light, what happens that whatever the starch that is present in a guard cell, that is actually converted into malic acid. Now malic acid dissociates into the malate anion and the H ion. Okay. So during light, what happens? There is a photo-induced transfer of H ion from stroma to the thylakoid due to plastoquinone pump. Okay. So it results into reduction in the cations in the guard cells. So once the reduction will happen, what will happen? That need to be compensated because due to disturbance in the electrochemical gradient, and that compensation is actually being done by the transfer of potassium ion from the subsidiary cell to the guard cells, right? And this event is actually called as an influx of potassium ion. Okay, so the moment potassium ion comes inside the guard cells, what they will do? They will bind with the malate anion, okay, and as a result potassium malate is going to be formed. Calcium also comes along with the potassium just to balance the electrochemical gradient, right? The moment potassium malate is formed, you know, potassium malate is a salt. Since it is a salt, what it will do? It will increase the osmotic potential of the guard cells, right? And as a result, there is going to have an endoosmosis. So once the endoosmosis will happen, the guard cell will become turgid and the stomats are going to open. So that is the thing that is going to occur in the light, right? Let's see what is going to happen in the dark. We know very well that in case of dark, there is an increase in CO2. Why? Because there is no photosynthesis. Okay. Then in dark, the plastoquinone pump is not going to work. So definitely the concentration of H ion within the stroma is going to increase. So as the H ion increase, the H ion will be transported from the stroma to the guard cells. So definitely here the potassium malate will break, and as a result malate anion is going to form. So malate anion will bind with the hydrogen ion and form malic acid is going to be formed. Now the moment malic acid is formed, you know very well that the osmotic potential of the guard cell is going to decrease. So this will result into the efflux of potassium ion, that is the potassium ion will start moving from guard cell to the subsidiary cell, okay, and this will eventually lead to exosmosis, and as a result the guard cells is going to be turgid, the guard cell is going to be flaccid, and if it becomes flaccid, then the stomats is going to be closed. So this is how through the active potassium ion theory, the very movement or opening and closing of the stomats has been explained, right? One more thing, there is one hormone that promotes stomatal opening, and that is cytokinin. And why? Because we know very well cytokinin is known for mobilization of the nutrients, and we have just seen that it is the potassium, that the very movement of potassium in and out is actually causing the stomats to open, and since cytokinin has a capacity to mobilize the potassium, that's why cytokinin plays a very important role in opening of the stomats, right? So let's talk about what are the factors that is responsible for the influencing the rate of transpiration. You know, we can divide the factor into two categories, what we call external factor and internal factor. But for all practical purpose, it is the external factor that has more significance. So we will concentrate here only the external factor. So the very first external factor is what we call humidity. Now what is humidity? Humidity is nothing but it is the amount of moisture that is present in the atmosphere. So greater the amount of moisture, the lesser will be the amount of transpiration. So we can say that rate of transpiration is inversely proportional to the humidity, right? Similarly for temperature, what will happen? If the temperature is going to increase, the metabolic activity is also going to increase, and thereby the rate of transpiration will be also increased. So for temperature, it is directly proportional. For light, we know very well that light, or particularly the blue light, actually it generates temperature, so we can say that blue light is also going to promote the stomatal opening. Okay. Then wind velocity. Now we have to understand the wind velocity. There can be two categories in which we can keep the wind velocity. One, the wind is gently blowing, and second, there is a very fast or windy environment. So if there is a very fast windy environment, what will happen? That air is going to have a friction with a leaf. In that case, the stomats are going to be closed, so rate of transpiration will dip. But suppose the wind is gently blowing, then what will happen? In that case, the rate of transpiration is going to increase. Because you see, if the wind is gently blowing, just above the stomats, it will create some sort of vacuum. Okay, and as a result, the water from inside will come towards the outer side. So that's why it is going to promote the rate of transpiration. Right? Atmospheric pressure, higher the pressure, lower the rate of transpiration. So that is the simple relation between the rate of transpiration and the atmospheric pressure. And availability of water, it's very clear that if the availability of water is less, the rate of transpiration is less, because eventually it is the water from the soil that is being transpired by the plant. Right? So this is all about the factors affecting transpiration. Let's discuss that how we measure the transpiration. The transpiration is actually measured by an instrument called potometer. Okay. Now there are three types of potometer. What are they? Gannong's potometer, Farmer's potometer, and Bowe's potometer. Now how we can differentiate? Basically, it is the design through which it can be differentiated, and there is one more point on which the differentiation can be made. For instance, in case of Gannong's potometer and Farmer's potometer, what happens? We insert air bubble in their marker. But in case of Bowe's potometer, there is a oil that is going to act as a marker. That is the only difference. Otherwise, there is a minor difference in terms of their design. There is one more method by which transpiration is measured. That is cobalt chloride method. So cobalt chloride is just a paper. Okay. So what happens just in a cobalt chloride, when it reacts with a moisture, then its color changes. Okay. And the intensity of color is equivalent to the total amount of water that is transpired. So this is the cobalt chloride method. Right? One last thing, guttation. What is guttation? First of all, guttation is the loss of water in the form of liquid, right, from the aerial portion of the plants. Okay. And that is going to occur through a aperture, what we call hydathode. Now mind it, hydathode is just like, it is not like a stomats. Because stomats can open and close, but it always remains open. So that's why it is not like a stomats. Just below the hydathode, there is a cells called epithem. Mind it, epithem may or may not be present. Okay. And it is present just above the extension of the xylem tissue. Okay, you can see there. So whatever the water that is coming from xylem, they are released from the hydathode in the form of droplets. Guttation is primarily observed in case of herbaceous plant, not the bigger plants. And the reason for the guttation is the root pressure, because as a result of root pressure, what happens? Excessive water is actually being pulled. So those water, suppose if there is going to have some atmospheric condition in which the rate of transpiration is low, then the other method through which the water can be taken out from the plant is the guttation. Right? There are certain factors that affect the guttation. What are they? Of course, they are humidity, soil water, atmospheric pressure, and temperature. So just think, if the humidity is high, what will happen? The rate of guttation will be high. Why? Because if the humidity is high, rate of transpiration will be less. So plant doesn't have any area through which the water can be expelled. Guttation is the way. So that's why under high humidity, what happens? Rate of guttation is high. Now soil water, directly proportional. Greater the amount of soil, greater the amount of guttation, right? Atmospheric pressure, again, in case of atmospheric pressure, what will happen? You can correlate with a transpiration. Higher atmospheric pressure, lower transpiration, thereby the rate of guttation is high. Okay. In case of temperature is high, then of course guttation is less. Why? Because temperature is high, rate of transpiration will be high, right? So naturally the guttation will be less. Okay. One simple thing what you have to keep in mind, that cloudy night always promotes the guttation. Cloudy nights means that humidity is relatively high. So when it is a high, then definitely guttation will be promoted. Okay. This is all about the guttation. One last thing, lenticels, because it also contribute in the rate of transpiration, right? Now what is lenticels? It provides means for the exchange of gases, but at the same time it consists of both dead and the living tissues. What are the dead tissues? Dead tissues are the cork cells, which is the dead tissues made up of the suberin. What are the living tissues? The name of the living tissues, what we call it, is a complementary cells. So it is a combination of both dead as well as the living cells, right? So this is all what I have to discuss as far as this session is concerned. Thank you.